Hello everyone. Today we are going to study the molecular basis of inheritance. In this video we will mainly focus on the structure of DNA, and in the next other ones we will proceed this topic. This video I am making from my handwritten notes. If you want me to make the presentation in form of typed form, then please message me on the Instagram. I have added my Instagram ID in the last of the video. I will make the typed form in the typed form from next time. Okay, let's see molecular basis of inheritance. In this chapter I make the notes from the NCERT class 12th. I added some words like the putative and the culminating to make you understand the NCERT. So in putative means supposed, and the culminating means the highest development. According to the highest development, it is supposed that the DNA was the genetic material that time. According to the scientist, in the 80s, DNA was the genetic material. It was supposed, not the proved. Not proved that time that DNA is genetic material or some other biomolecule is genetic material. So DNA and RNA both are the genetic material. The RNA have the different functions. It act as the messenger, adapter, structural, and the catalytic molecule. How it act as the adapter? In the protein synthesis. In protein synthesis, RNA act as the adapter. Everyone knows the RNA have three types: mRNA, tRNA, and rRNA, ribosomal RNA. So the tRNA function as the adapter molecule. It translates the 3 nucleotide codons sequence in mRNA into the suitable amino. In this chapter we will see structure of the DNA, process of making the RNA into DNA, in the protein synthesis means the translation. The 3 processes: replication, transcription, and the translation. The DNA is polymer of deoxyribonucleotide. Polymer of deoxyribonucleotide means the pair of nucleotides make the base pair. One nucleotide and the other nucleotide, it make the base pair. The length of the DNA we can define by the number of nucleotides or the number of base pairs. Means how many number of nucleotides are present in the DNA will define the length. Example, the bacteriophage. Phase into 174. This is the bacteriophage, and it contains the 5386 nucleotides. 5386 nucleotides are present in the bacteriophage phase into 174. The bacteriophage lambda contains the 48502 base pairs. This defines the length of the genetic material. The gene E. coli contains 4.6 into 10 raised to power 6 base pair, and the human DNA is 3.3 into 10 raised to power 9 base pair. The structure of polynucleotide. The structure of polynucleotide. A nucleotide contains the nitrogenous base, pentose sugar, and the phosphate group. These 3 combine and form the nucleotide. The nitrogenous base is divided into two parts. One is purine and the second is pyrimidine. The purine contains adenine and the guanine. The pyrimidine contains cytosine, uracil and the thymine. Uracil is used at place of thymine in the RNA. Thymine is present in the DNA and uracil is present in the RNA at place of thymine. So, adenine, the nitrogenous base and the pentose sugar attach and form the nucleoside. When the nitrogenous base, pentose sugar both form the nucleoside, and this nucleoside when attached to the phosphate group, this phosphate group attached to the nucleoside and form the nucleotide. Okay. The nitrogenous base and the pentose sugar is attached with the N-glycosidic linkage, and the nucleoside and the phosphate attached with the phosphoester linkage at 5-OH. What are the nitrogen bases? These adenosine, guanosine, cytosine, uridine are the nitrogen bases. The uracil is present in the RNA at place of thymine. So the two nucleotides are linked through the 3,5 phosphodiester linkage. Two nucleotides means the group of nitrogen, phosphate and the sugar. This one group attached with the another group of nitrogen, phosphate and sugar with the phosphodiester bond. The polymer has at one end a free phosphate moiety at the 5 end of the ribose sugar, and the 5 end of the ribose sugar to the 5 end of polynucleotide chain. Look into the diagram how this phosphate group attached at the 5 end of the ribose sugar. This is the ribose sugar, pentose. At this 5 end, 1, 2, 3, 4 and the 5, this 5th end, this phosphate group is attached here. Okay, this phosphate group attached at the 5th position, and this 5th and this chain form the polynucleotide chain. Please share, like and subscribe.